So this was a study that was conducted in collaboration with Dr. Jens Kuhle and his research group at the University Hospital Basel. This is a long-standing partnership we've had with them, and we've run many studies with them over the years that have been reported previously at ACTRIMS and ACTRIMS. Disease progression is currently measured in a mostly qualitative way using clinical and radiographic evaluation. So we were seeking to evaluate progression from a biological perspective using serum proteins. MS is a very heterogeneous disease, and disease progression is very complex. There's various subtypes and phenotypes of disease progression. Furthermore, it typically manifests over longer periods of time versus disease activity. There's been a great deal of focus in the field of late on the concepts of PIRA and RA. So PIRA being progression independent of relapse activity, and RA being relapse-associated worsening. In this study, we sought to identify biomarkers that were associated with radiographic indicators of PIRA. And thanks to our collaborators at University Hospital Basel, we were able to access a very carefully curated set of samples from 37 patients that represented two carefully matched phenotypically extreme groups, worsening progressive MS and then stable MS for comparison. And notably, the patients with worsening progressive MS had no relapses during the follow-up period, and so therefore they represent pure PIRA. We also analyzed additional assays that are available using the Olink proteomics platform. In total, we measured almost 3,000 protein analytes, and we used NfL and GFAP measured by Simoa, as well as our custom assay panel. These are two biomarkers for which there's a lot of focus in the field as disease activity and disease progression biomarkers. We use those as benchmarks to evaluate associations relative to both gray matter volume and white matter volume. These are two MRI endpoints that are associated with disease progression. We had previously reported associations of our serum protein biomarkers with clinical endpoints of progression at ACTRIMS in October 2022 from the same data set. So after running the assays, we applied stringent analytical QC to the proteins, and we were left with just over 2,000 analytes. And we found more than 60 proteins measured at baseline that had prognostic potential to predict gray matter or white matter atrophy that were as significant or greater than NfL or GFAP. Interestingly, the largest effect size were seen in proteins that are primarily expressed in the CNS. This is an important aspect of the work we've done at Octave, which is focusing on biological corroboration of our findings. So whereas it's important to establish statistical association with the endpoints, it's also critical that we understand the rationale for why that protein was associated with MS. In other words, what cell types, what pathways, and what mechanisms are involved. Work is already underway to verify these findings in additional cohorts. And additionally, we want to investigate multivariate algorithms that leverage multiple proteins to potentially enhance the signal. Similar to what we have validated for disease activity using the MSDA test, we believe that a multi-protein approach will have stronger significance for disease progression endpoints than any individual protein by itself. MS and particularly disease progression are complex and heterogeneous. Developing accurate endpoints that allow us to predict the likelihood, the extent, and the rate or velocity of progression is still very much an unmet need in MS care. Right now, disease activity is better controlled with modern DMTs versus disease progression. So addressing disease progression is in some ways the next frontier in MS therapeutics. I think the two most important benefits are, one, establishing accurate and precise measurements of disease progression for use in clinical trials to evaluate efficacy of DMTs specifically related to PIRA, progression independent of relapse activity. And number two, clinical use of these tools to enable a personalized medicine approach in which the right DMT is selected for a patient based on their future risk of progression. It's likely that finding the right endpoints for these purposes will require a multimodal approach. So by integrating biological biomarkers, neuroimaging, and clinical measurements together, that's really the Octave approach. And we have programs in each of these domains that we are working on and several that are commercially available.